So my grant was in 2011, and it was looking at the SETD2 mutations in kidney cancer. And my mentors were Eric Yonash, Nizar Tanir, and Minchi Hung. And this was a great team because you essentially have two great clinicians working with the basic scientists. So literally, I would see patients in clinic, walk across the street, and go to the lab. Essentially what I was looking at is one of the more rare mutations in kidney cancer called SETD2, and looking to see how these mutations in SETD2 change our chromatin structure or the rearrangements in our DNA. So as a fellow coming out of fellowship, I was going to start my own laboratory, and when the TCGA or the Cancer Genome Atlas identified all these mutations, I intentionally picked a mutation that was pretty rare because I thought, well, everyone's going to go for the more common ones, I should go for the less common one. So essentially, during that time from the KCA's grant, I essentially identified some tools that we could use in kidney cancer to basically re-engineer the same mutation that we see in patients and put those in cell lines. So I used that money from the KCA to build some tools and also show that when you lose this SETD2 mutation, or you have this SETD2 mutation, you lose a very specific mark inside the protein of the DNA. And as part of that, that actually led to essentially 2 Department of Defense awards as well as 2 R01 grants, which continues to fund my lab to this day. Yeah, so as a physician scientist, I see patients, I also work in the laboratory, so it's important for me to do research that's relevant to the patient, and I want to see it happen during my lifetime. So one of the things that I identified with SETD2 mutations is using the Mayo Clinic database. We actually looked at 20 years of tumors, and we identified that SETD2 mutations, when present, are actually associated with the worst prognosis. So when we correct for clinical prognostic algorithms such as the Mayo Clinic sign score, so this incorporates things like stage, size, grade, necrosis, these are things that you can pull from a pathology report. And when you incorporate that prognostic score, it gives you a sense what the risk of recurrence is. And this is often used in decisions for treatments like adjuvant therapies, like immune therapy, for instance. So using that sign score, I found that these mutations actually add more prognostic value. So you can actually identify more patients who are at higher risk. And as a result, you can actually potentially identify patients who are more prone to improve from immune therapy or reduce the risk of recurrence. So that was sort of the first phase of my career at Mayo Clinic. So the second phase is looking to see how these mutations change the tumor microenvironment. And one of the things that our lab discovered in combination with Dr. Keith Robertson is that if you take a SETD2 mutant tumor, it can actually make its neighbors look worse. So essentially it signals its neighbors to do something even worse. So it's sort of like having a bad actor in a classroom, and that bad actor essentially influences the other surrounding students to act poorly as well. And this is sort of interesting because we know that tumors are heterogeneous, and sometimes SETD2 mutations may be kind of rare at first, but they get progressively enriched as the tumor becomes more aggressive. This suggests that there is some influence of these mutations on the tumor microenvironment where it can almost subvert the rest of the tumor to more aggressive variant. So anytime you have a mutation, it can act as 2 different markers. One is what we call prognosis, where irrespective of what we do, the tumor is going to do very poorly or it's going to do very well. And then we have predictive markers. These are markers where, when present, we have drugs that are available. So currently, I would say that SETD2 acts as a prognostic marker. We don't have any drugs specific to target that, but this is sort of the next phase of my career. Is now we've identified sort of the mutations. We identified what they can do to influence disease recurrence. We have identified how they can sort of influence the other neighbors. So the next phase of the career is actually try to develop a drug. And it's challenging for this type of mutation because it's what we call a loss of function mutation, which means the gene stops working. All our treatments in cancer typically target things that are overactive. So if it's overactive, we can use monoclonal antibodies, we can use small molecules, and that shuts down a hyperactive signaling pathway. But for this is a loss of function, which means it's turned off. So we don't have a lot of drugs to turn that on. But this is something that we're currently actively working on, where we're trying to identify from the lab, are there some pathways that are hyperactivated as a result of a SETD2 mutation that we could target? So when we think about different grants, I think that often we'll try to go for the big grants, but the big grants require a lot of preliminary data, where smaller institutional grants are actually good because you can take more risk with those grants, and you can also use that to generate preliminary data for a cancer grant from the NCI. Typically when we think about pay lines from the federal government, we're looking at 7th to 9th percentiles for funding. So that means that the majority of grants are actually not funded. So I think that these grants are actually helpful for young investigators to sort of compete with the larger group of physicians, and particularly for physician scientists who spend their time seeing patients, they may be competing against full-on PhDs. So I think it's important to have these grants that kind of help take a little higher risk, and it also uses it to get some preliminary data for these larger grants. So as a PhD, I actually worked in a rare genetic disease called myotonic dystrophy. And as a PhD, you're in the lab quite a bit, you're doing a lot of wet bench work. But I sort of had my first foray into patient advocacy, meeting patients who were affected by myotonic dystrophy. And one of the things that impressed me is when you shake their hand, you can tell that there's already something different about their handshake. And I think that sort of hit me that, oh, well, this is something that really touches people. Sometimes when you're working in the lab, you don't see that aspect of it. So I think being involved in the patient advocacy programs, whether you're an MD or a PhD, actually keeps that human perspective. As a result, when you see that human perspective, it helps you pursue some of the topics that might be relevant to patients, not just relevant to basic science.